Lights, southwest corner door.
This is east, west, south.
Southwest?
Yes.
I can tell you where southwest is.
Yeah.
All right. I'll give you a quick introduction.
Okay, thanks.
I'll do a better job than I did yesterday.
I did a master's in Berkeley, not a PhD.
Okay. Hello, everybody. First, I'll introduce myself. I'm Preston Manwaring. I'm actually a professor of computer engineering and neuroscience down at BYU. I know, no groans. And I have invited a good friend of mine from University College London, and he does very interesting research in electrical impedance tomography. He is a neurophysiologist. He did, so he has an MD PhD now?
Yes.
MD PhD, did his master's at Berkeley, has been working in the monitoring and imaging space for neurons for quite some time. I had a PhD, he was on my dissertation committee many years ago. Anyway, I wanted to invite him down here to show him what the University of Utah is doing and networking opportunities. And I hope it's a very exciting confluence of electrical engineering, computer science, and neural engineering. So I guess that's what you guys call that, is neural engineering. So I don't have that. So that's wonderful. All right, so I'll turn the time over to.
Thank you. Thank you. Good afternoon. Should we get rid of the teams? That should be good. Right. Oh, sorry, I've got to go back. Good afternoon. Thank you for coming to listen to me. This is a great opportunity. And I had a crazy idea a long time ago, which is sort of working. And I'm looking for collaborators. I hope I don't want to tell you more about it than you want to know. But I'm going to go through the technique. It's a nice small audience. Why don't we just make this interactive? If anything, I think I lost a few people yesterday when I gave a talk in BYU. So if anything isn't clear or any questions, why don't we just make it interactive and talk to me as we go through? But I changed the drift slightly. I take it that the audience is basically biomedical neural engineers. So let me pitch that right. Good. So I'm David Holder. I'm a practicing physician doing EMG and EEG at University College London. There's a hospital associated with the university, but I've also got a bioengineering and neural engineering interdisciplinary research group where we've been working on this for quite a while. So this is the background. This is why I, as a neurologist, have been pursuing this crazy idea for a long time. So I probably don't need to tell you this. There's this marvelous book on networks of the brain, a little bit old now, from Olaf Sporns, that what a lot of research tends to be driven by the technology that's available rather than what we really need. And so there's been marvelous advances, which I don't need to tell this audience about. But the way the brain obviously works at cellular level with small networks of dozens or hundreds of neurons doing computation. But as a neurologist, what you see is that they're the major pathways in the brain, which are also essential to understanding most clinical conditions. And there's this, I won't read this out for you, but I think you can't understand the brain without understanding what is probably a few score of major anatomical and functional pathways in the brain. And the bottom line is, in spite of fantastic advances in neurotechnology over the past 2 decades, there still isn't any method that allows us to image circuit activity in the brain at what I would call probably mesoscopic level of milliseconds and say a few millimeters in the brain. So what do we have? Well, we have electrophysiology is very advanced. We've got neuropixels we record from hundreds of contacts. It's high time resolution. Providing you're on top of the local field potential, you get a very good signal to noise, but it undersamples vastly. We've got 10,000 billion neurons in the brain, and even the neuropixels is only able to record from a few hundred of them. This can't possibly allow us to sample the network activity. And if you try to do modeling from it, there's this technique of inverse source modeling of the EEG that can be used, but you have to make numerous assumptions, and if they're violated, you don't know how accurate the technique is. So what I want to talk to you about today is what I've come up with. I came up with it in the library in Berkeley, as Preston mentioned. I had this idea as a young doctor, and I decided I knew what I wanted, which was a cross between CT and EEG, or evoked potentials. Everybody told me it was impossible. When people tell me things are impossible, that just redoubles my desire to do them. I sat in the library in Berkeley for a year, went through every possible method, X-rays, ultrasound, microwaves, and I came up with this idea that the biggest change, that everything in the brain that changes is tiny, except one thing, which is the membrane resistance when you have an action potential, which changes by 80 times. And at that time, people were just talking about this technique of electrical impedance tomography. So I thought I was mistaken. I thought I spent 5 years making this machine that can image all the circuit activity, and the rest of my career doing computational neuroscience. It hasn't quite worked out like that. I want to tell you about this fascicular EIT, which we have got working. So the advantage of EIT is there's a unique inverse solution, unlike inverse source modeling. And we've shown that in the rat cortex, you have a resolution of 200 microns in a millisecond. The dream was, this is the bottom line, the dream was that we would be able to do it with EEG electrodes. You inject a cut, I'll explain it all in a moment. And so you'd have something looking a bit like a cycle helmet, and then you would show someone a picture of their grandmother. You would be able to see all the circuits lighting up over milliseconds. The bottom line is we haven't been able to do that because the signal to noise is diminished by the skull, but it works with intracranial electrodes. But we can also do a second mode, measuring a different impedance in a different way, and we can image in real time slow changes, a bit like having someone in an fMRI scanner. And here, this is what an EIT system looks like. This is what the images look like. Whoops. This is what the images look like. Okay, so that's the background. So what is electrical impedance tomography? If you're a BME, you might actually have heard of it. You might have been lectured on it, but it's very small print, new medical imaging method, and it's mainly been pioneered for imaging in the lungs. So it's been around since the 1980s, and the main application is you put 16 EKG type electrodes around the lungs, breathe in and out, and you can see a fuzzy image of the lungs breathing. And that's been its main application. What we've done in my group at University College London is to adapt that for imaging in the brain. So the underlying principle, this is a current source. You apply a current. You can apply current to any combination of electrodes you like, but usually it's to one pair at a time. You measure all the other voltages. Normally it's done at kilohertz, and that gives you lots of these transfer impedances made with these electrodes. And then you can reconstruct them into an image. This is what an EIT, our current EIT system looks like. It's quite conceptually simple. We have a Keithley, very high spec, very low noise current source. Then we have a high spec EEG amplifier, 128 channel Actichamp EEG amplifier, but you need 24-bit A to D because you've got a big carrier signal. We're looking for very small changes on the back of that carrier signal. We make our own multiplex and written our own software, but that's all you need. That's a tank we've constructed for testing. And the principle is that during the action potential, I mean, I'm sure you all know this, you have ion channels in the nerve, where as you get a voltage gradient change, they open and the impedance changes. And in fact, this is how the action potential was first discovered by Cole and Curtis in 1940 because of this big impedance change. So if that's happening locally in a neuron, when you have activity in a part of the brain, it's going to cause an impedance change. And that then you could image with this EIT. And this again is an example of what the image looks like. The technology is that you would have 128 electrodes over a rat cortex. You do a craniotomy, place it on the brain, have some sort of repetitive trigger, which could be somatosensory evoked response, it could be whisker stimulation. But the important thing to realize is that looks like an fMRI, but the timescale is milliseconds. We can image over milliseconds with an accuracy of a fraction of a millimeter. So the background to EIT, there's it's a moderate it's a population of groups working on about 40 worldwide, mainly interested in lung ventilation. There's 4 companies now who are actively marketing it for this. And I think that my prediction is that it's something that can run at the bedside. And I think in 10 or 20 years' time, every ITU will have the part of their monitoring. They'll just have a band of electrodes on and you'll just see the lung ventilating. That's not something we've done much research on. And then this is just showing one of the things from one of the companies. And here's an example of its use, not lungs. You put a ring of electrodes around the stomach. You drink salty water, that changes the resistivity of the contents of the stomach. You see an image like this and you can watch that extinguish over a few tens of minutes. Does it actually work? Yes, but it's a soft field inverse problem. So the way to think of it is that if you've got, so you're doing it in the head, you inject a current and record a voltage. That's a 4 electrode impedance measurement. What you're sensitive to is it's not like X-ray where you've got a very narrow beam and hence the accuracy of X-ray CT. If you can conceptually, it's like having you're sensitive to a banana-shaped bit of tissue between those electrodes. But when you collect lots and lots of these banana-shaped bits of tissues and then do clever maths on it, you get an image. But this together with, as in you might be familiar with NIRS and photic imaging as well, when you do it with light, that's a soft field problem. But this is it's an old paper, but this is just to give you an idea. When I gave the talk yesterday, I think one of your students didn't understand what exactly we were doing. You have a tank of salt of water, you put a cucumber in it. As you move the cucumber around, it's not crystal sharp like it would be in, say, X-ray CT or MRI, and it's a little bit blurred, but it's a similar resolution to radioisotope imaging, and it actually works, and it's a unique inverse solution. So what I'd like to tell you about today is firstly to introduce the actual. Yeah, go on.
What is the spatial resolution?
The spatial resolution is about 5% of the diameter of the image. So you put your electrodes around the edge of it, it's roughly 5%. So what I want to talk about is introduce the technique to you to give you a picture of what's actually involved. And then with my traffic light system of what's wonderful and what's worked, is there's 3 topics just to go over. The most important one, the one which I think is the future of it, is in brain. And that's partly worked. But as I said, we haven't got the pot of gold at the end of the rainbow, which is the cycle helmet. But at the end, I want to tell you how we're working on it. And I think we are going to achieve that. And then there's a couple of projects that have worked. One is this fMRI brain thing. And then another one is, which has been very successful. And we've had DARPA and NIH Spark funding, is imaging in the vagus nerve, which I never dreamt of doing when I first started this. So I'll just mention that. So what's the principle? The principle is, if you're recording impedance across an axon, it's got these ion channels in. If you apply a current at the frequencies we use, which is under about 100 kilohertz, it's effectively it's quasi-static. you don't have to use Maxwell's equations for capacitance. So the current will travel in the extracellular space and go between the cells. Then when the ion channels open, the current can now go inside the cells. There's more salt inside the cells, and you get a small decrease. It's surprisingly small because the extracellular space in the brain is only about 10 or 15%. But when you look at the biophysics of it, it means that at DC, it's about a 1% change. At the frequencies we've got, it might be roughly that or 10 times smaller. Now, this is where it starts to make your brain hurt, so bear with me, but it is quite important, and please ask questions if it's not clear. When we originally did it at DC, which is where you should get the largest change of this 1%, it didn't work because we're drowned out by the brain's own EEG. So if you're measuring at DC with, say, a bandwidth of up to 100 hertz, that's where the EEG is, and you were just measuring a potential on top of our carrier, and you had to average for a very long time. So we had this great insight that if you go up in frequency, although the signal falls off, you get the noise falls off a great deal more, and there's a sweet spot. So this is just illustrating it. The problem is that when you go up in frequency, the signal's smaller because you've got these very thin lipid membranes in all your nerves, and they've got very high capacitance. So even at a kilohertz, some of the current crosses the capacitance and goes intracellular. So therefore, when the ion channels open, it's less of a difference. It's about 10 times less. That's illustrated here. But we discovered that in the brain, we have a sweet spot at about 1.5 kilohertz. Now, so that's the signal to noise. This is kilohertz along here, up to 10 kilohertz. That's the actual size of the change. Now, the important technical issue is we want to measure millisecond changes. So this is a bit like AM radio. We've got a carrier at 1.5 kilohertz, but you need a bandwidth of 500 or 1,000 hertz either side of that in order to be able to get that time response that we want per millisecond. So this means that in this mode, we're constrained. We can only measure one frequency at a time because we're at 1.5 plus and minus 500 or 1,000 hertz. Does that make sense? Yeah. So if you're in telecommunications, I think this is bread and butter for you. So then this is illustrating what our technique is. So if you can imagine you've got the rat and you're twiddling its whiskers, or you've got a somatosensory evoked response, and that's illustrated here. This is the potential you're measuring because of the evoked response. This notionally is the impedance change that will be associated with it. This is just diagrammatic. It's not that big. It's about 0.1%. And this modulates the carrier. So you're applying a current like this, but if it's in tissue, which is being active every, half a second, again, this is exaggerated for explanation purposes, you see a modulation of that signal. And then what you actually record is a combination of the two of them, which combines the EP with the modulated carrier. And then you will record that, say, for 32 repeated evoked responses. You concatenate them into just one epoch, and then you can filter it, get out the evoked responses, and demodulate it, and you get the impedance response. This is an old slide. We do it a slightly different way now, not subtracting it. So what kit, I've already mentioned this, current source, high spec amplifier, which I said is 24-bit A to D, because we've got a big carrier and a 0.1% change on it. We make our own multiplexer, and then we also need to make our own electrodes. So over the course of developing this technique, there are lots of areas where we thought we could just use commercial equipment, such as electrodes. But it turns out that in order to do the impedance measurement,
I'm sorry, this is taking you back a slide, but is it, you have to use the same precise stimulation or evoke the same response with each input in order to separate the evoked response and the evoked.
Yes, we have to
response?
assume stationarity. So we will do an evoked response over, say, 10 minutes. The assumption is the brain's response to that evoked response. For example, just like an evoked response in neurophysiology, we're assuming the brain changes in the same way each time. And then we average those together over time with just one current injection in order to get a decent signal to noise. Then we move, I'll explain this in a moment, then we move the current and repeat that and repeat that. So this is one of the limitations, that it takes 10 minutes of repeating something such as an evoked response. And then we make our own, so we couldn't use, it would be great if we could just use neuronexus electrodes or other subdural electrodes, but we make these ourselves, which are made from platinum or stainless steel foil, and we laser cut them on silicon rubber. And then this is just showing us the electrode design. The idea is that we want to have 64 electrodes on each hemisphere of a rat brain. And the geometry is that they're 0.6 of a millimeter across, and then we design our tracks, and then we cover them with P-dot, which is the way of reducing the electrode impedance. And that's what one of them looks like. And it's great having this facility in our lab because we can change the geometry depending on what the application is. So this is just to go over the imaging, which is different to the way imaging normally takes place. So just bear with me. So if you can imagine, you've got this array on a rat's brain. You apply the current at 1.5 kilohertz, and you will apply it to those two electrodes, say for 30 seconds. During that time, you keep repeating an evoked response or twiddling the whiskers. And then what you will see when you measure the voltages from these other electrodes around is you will see a change in the impedance relating to the activity, but it's a sort of combination of how much that brain is active together with how much current is going. That will be another, and then you move your current to another pair, repeat the same thing, and keep going round, and it's about a quarter of an hour to get the whole data set. Though you take all those measurements, we use a finite element model of the brain in order to do some advanced maths, which I haven't got time to go into today, but it reconstructs all these transfer impedances into an image that looks something like that. But the important part to note is that you get an image every millisecond. So here's a typical experiment. This would be in, this is in the rat brain. We anesthetize the rat. We would remove the skull. We put our electrodes on the skull. We have a repeated, in this example, a 4-pore sensory response. This is the technology. And then you will consecutively apply current to about 30 pairs of electrodes. And then recently we're collaborating with a German manufacturer of impedance kit, so we can have all our EIT hardware in just one box, which is going to be available for collaborators. Then, if you can imagine, this is just diagrammatic, you're measuring for 15 minutes, and on each you've got a few hundred sine waves with a modulated signal, lots and lots of them. Of course, they're all different sizes. The carrier are different sizes in different places. And then you low pass filter them in order to get the evoked response from which will be different in different electrodes. And then what you see is you then get the impedance change, which is different in all the different combinations of electrodes like this. So there'll be one spot where you've got the combination of the largest current, but also the largest technical change. And then you take all of those, and then this is what, I think I've already shown you this. This is the image. So just to be, that's the method. So the important thing to note here is that this is over milliseconds. We're looking at a change in S1, in sensory cortex, and you see the change start and build up, and you look at the trajectory over milliseconds and over different layers in the cortex. So that's so that's explaining to you the introduction to the method. Now I want to talk a bit about what we can do with this. Sorry. Yeah.
If you do a more complicated task where you might have an activation somewhere, and inhibition somewhere else, could you find based on changes in the impedance and the extracted waveforms, like inhibitory and excitatory neurons?
It's a great question. So what we can tell you is if there are changes in the electrical impedance. Now, if there's inhibitory changes, that will cause a decrease in impedance as well, because you can only get a decrease in impedance. So we wouldn't be able to distinguish as to whether it's excitatory or inhibitory, but we will just tell you whether ion channels are opening, but you can't distinguish between them. Yeah, so it's a nice question. So now I want to tell you just a few examples of what we've done in brain and where we're at. So here's another example where we produce epilepsy in a rat model. We provoke a seizure by electrical stimulation, and we've got an anesthetic, that alpha chlorelose, that allows the seizure to propagate. Then I assume in this audience most people are familiar with ECOG, and so this is the seizure propagating over seconds, and these are spike waves. Remember that in order to do this, we have to trigger off a repeated change. So in this case, we trigger blowing this up, we trigger off these spike wave complexes, then you have to repeat the seizures, and you get them for all different possible current combinations. And then you end up with an image that looks like this. And now we're seeing something with that same resolution of milliseconds and 200 microns in the cortex, but you can watch the seizure start and spread. So the dream is that we can do this in humans and be able to look at the propagation of activity during epileptic seizures. Here's an example of how we can, with this resolution,
you're measuring like the sequence activity learned, right? But to that extent, like what's the smallest amount that you can actually measure? Because you're measuring like how mild of a seizure, kind of things like that. Would you be able to actually still measure?
Okay, so signal to noise is a problem, which is why we have to average repeated things, say, normally for 15 or 30 seconds. So all I can tell you is what we've measured so far, which is we measured during physiologically evoked responses, which is with somatosensory evoked responses and visual evoked responses, and also whisker stimulation. So that's normal activity. And then we measure during epileptic seizures. Now, epileptic seizures is paroxysmal, activity. We haven't done the studies for something more subtle or cognitive paradigms, which may give a smaller depolarization. So the answer is we're not sure. All I can tell you is it works under those existing paradigms. Yeah, but the signal to noise is pretty good by the time we've done that, and with this paradigm. So our signal to noise is 20 to 30 to 1. So there's quite a lot of overhead if we want to measure something more subtle. We just haven't done it yet. So the next question is, how deep in the brain can you image with subdural mats? Because the original dream was it was tough not being able to do it on the scalp. But I thought originally when we started, just being an optimistic doctor, that when we put subdural mats on, we would be able to image everywhere in the brain. And the dream was you could watch activity coming up through your spinal cord, brain stem, and then the trajectory. You can't image everywhere in the brain because you're limited by the amount of current you can put in, because we're very careful about that. You mustn't put in enough current such that it starts to modulate the brain function. So, but what we've shown is that with evoked responses with a subdural mat, we're sensitive down to about the outer quarter of the brain. And then here's an example of where we had a hippocampal with epilepsy. So what we did was we recorded the activity in the hippocampus, the spiking in the hippocampus. And with that, we were measuring with mats on the surface of the brain. And because the epileptic activity is larger than the evoked response activity, we could clearly, and this is all published, we could clearly see these seizures in the hippocampus, which is a good sort of quarter to a third of the way into the rat brain. So we can see deep, but we probably can't see as far as the brainstem unless we improve things. And on that note, I don't know if you know Ken Shepard at Columbia, but we're collaborating with him, and we want to use Neuronexus type electrodes in the brain and do EIT. And with those, we ought to be able to see everywhere in the brain, but we haven't done that work
There's
yet.
an old paper by Isaacson, right, 1980s.
Sorry.
Yeah, I think there's an old paper by Isaacson in the 1980s that addresses this challenge when all the electrodes are on the outside.
That's right. He's my chum at RPI.
Yeah.
Yes, yeah. He's a dear friend. But he thinks he's a mathematician, so there's a whole thing about how best to do this imaging, and they were mainly interested in breast cancer screening at RPI. In theory, you get better images if you have sinusoidal patterns of activation and you apply your current through all the electrodes at the same time. And the maths and the theory tell you it does. In practice, it doesn't. The best method in our experience is one pair at a time. But it may be with improvements in instrumentation in the future, his approach would be better. So then, for the electrophysiologist in the audience, the obvious question is this any better than what we can already do with electrophysiology? And the answer is it swings and roundabouts. The signal to noise is better in ECOG or measuring local field potentials, but only if you're right on top of the source, the moment you start getting away. And so that's why it takes us longer. It's not real time, which is a limitation. But if you're measuring just potentials, firstly, the fields can cancel. And then secondly, you're entirely sensitive to the orientation of the current source. If your contact is there and your current source dipole is pointing at the contact, you'll measure something, you're totally blind to it if it's tangential. Impedance doesn't suffer from that problem. You will measure the impedance irrespective of the geometry. So the bottom line is, and the other thing is you can't image, you can't reliably image voltages, whereas ours is a unique inverse. But I think the two techniques are complementary, and I think that this could complement making measurements of electrophysiology as they're done at the moment. So in summary, at the moment, the original vision was we could do it non-invasively with scalp electrodes. We can definitely do it, but at the expense, number one, of intracranial electrodes, either on the surface of the cortex and we're working on depth electrodes, but you also have to average to some sort of repeated stimulus over minutes. And then, how are we for time? I think we're okay. So we're working on being able to achieve the original goal by making magnetic measurements rather than electrical measurements. I'll talk about that at the end. We're also working on having these penetrating electrodes, which we're working with Ken Shepard. And we're looking for collaborators to extend what we've done in the rat to human trials in epilepsy. We've got working kit that's human approved for that. And I'm working with this German company to provide, at the moment we collaborate with a number of groups, mainly in the US, because we've had US funding, but we have to send our own team over because we're the only people that know how to work it. We're simplifying it such that we can provide a kit and support it to collaborators so they would be able to do their own experiments. And I think it could be a useful technique for animal cognitive neuroscience studies. So now, Green, this has worked. This has been a great, unexpected success. So one of them is the idea of using EIT, but in what I'd call functional MRI mode. So it turns out that there's two different mechanisms that give you an impedance change in epileptic seizures. I've already mentioned the one before, but there's this one. So in epilepsy, you're applying your current, goes between the cells. They run out of energy and they run out of ATP. The cells swell over tens of seconds during the seizure because the cell isn't in osmotic equilibrium and therefore water moves into the cells. That decreases the extracellular space, and you get a big increase of about 10 or 20% in the impedance, which is much easier to see. And it's over tens of seconds. This is our, it's actually the same hardware I showed you earlier, but it's boxed up for use in a clinic. And we can make these measurements and it works. So here's a study that we made with a collaborator in a pig, but using human depth electrodes. And in it, you can see in real time, this was a model of epilepsy, a focal epilepsy model, and you can see the seizure onset zone, and you can see it spreading. The other advantage of this is that in the other technique, I mentioned that it takes averaging over tens of minutes because we have to go through all the different electrode combinations. Because this change occurs at much higher frequencies, it doesn't need that sweet spot of 1.5 kilohertz. We can we can activate all the electrodes in parallel by frequency division multiplexing, i.e. it's a bit like on your mobile phones, you have things going at different frequencies, so you have many channels. We can apply one frequency, say 50 kilohertz here, 51 kilohertz, 52 kilohertz, on all of them at the same time. So this offers a real-time way of seeing the seizure onset. It works in pigs. We haven't been able to do it yet for various practical reasons in a human, but I think that this offers a way that if you have got an epilepsy surgery telemetry unit, as you do here, it will be like having the patient in an fMRI scanner, but for the whole 5 days whilst they're being monitored in the unit and they're having their seizures. We've done a modeling study to show how EIT would confer an advantage over what's done at the moment. So what's done at the moment is that you put a number of, this is a diagram, but these are from real epilepsy patients. Patients come in, you stop their medication, the neurosurgeon puts about 10 electrodes in the brain, and you monitor over a week whilst they have their seizures. And at the end of that time, you see where you see spiking at different contact, you say that's where the seizure is coming from. Now, if you do, so we modeled this as if we were going to do EIT. So this is just a computer modeling study. If the bottom line is from the current way of doing it by making electrical measurement, is that if the seizure is within a centimeter of one of your contacts, it's localized very accurately by the existing method as shown here. And this is the an example of, just one example, we did 30 different seizures, and EIT is similar. It's actually EIT is better, but we were doing EIT measuring from these electrodes, but also scalp electrodes. But if you take an example where the seizure is away from any of your contacts, and the neurosurgeon have to guess where the seizure is coming from, and if they guess wrong, the normal way of doing it by measuring EEG is hopeless. You can't measure anything. If you try and do inverse source imaging, it's hopeless. EIT is much better. And we were able to show in this modeling study that if we were able to do this, this could be a real paradigm shift in the technology for localizing seizures in patients that have these intracranial electrodes. So my prediction and hope is that in 10 years' time, this will just be a routine complement to what is done at the moment of measuring EEG in these patients. So that's that. Then, I think we're okay for time. This is the unexpected project that's been very successful. So you have the vagus nerve in the neck supplies about 8 different organs, including your vocal cords, your heart, your lungs, and it's autonomic, so it modulates the activity. And for a couple of decades, it's been found that if you electrically stimulate it, it works in reducing epileptic seizures. Nobody understands why. I don't know how they. But the problem is these poor patients have got this thing in their neck, and every time it switches on, they can't talk because it's stimulating the larynx, but it can also have effects on heart and breathing and their digestive system. So we've collaborated with a pharmaceutical company, GlaxoSmithKline, which then spun out with Google Health, Verily, because they want to stimulate this vagus nerve, but they want to avoid the side effects. So the question is, could you do that? And at that point, unbelievably, so if you cut, I think yes, so if you cut a vagus nerve, I don't know how much you know about nerves and fascicles, but this is what I do with my other hat on. Here's an example of a nerve cut. This is a nerve to one of the limbs. And what you see inside it is that's the outside of the nerve, but you see what are called fascicles. So fascicles are a bundle of some hundreds or thousands of nerves, and they've got a perineurium connective tissue around them that helps control the extracellular state. And you see these fascicles. After the Second World War, where there were a lot of nerve injuries, there was a Australian pioneer who worked out what these fascicles were connected to, and it made sort of logical sense that you would have one fascicle for one muscle or a group of muscles or one area of skin. But when we started our project in 2018, nobody knew what these fascicles were connected to in the vagus nerve in the neck. So, oops. So we started, so that's just explaining about the vagus nerve and stimulation. And we wanted to know what these fascicles, were they connected to one organ, or did it make no difference? And if they were connected to one organ, could this then be a way of leveraging it such that we could do selective stimulation? So the idea was, everything that you do in EIT of the brain becomes easier if you're doing it in a nerve because it's a nice cylindrical structure. So we adapted our electrodes to be a ring of 14 that went around the nerve. And then the hypothesis was you'd see these different fascicles, and could we see if they were connected to a particular organ? And then you would, once you found there was one, for example, the heart, then the EIT image would light up like that. You would focus your current on the heart, and it may be able to help in heart failure. So this is what we call the UCL nerve cuff. There are lots of groups. I don't know if there's anyone in Utah working on this, but there are lots of different designs for nerve cuffs. Many of them are penetrating, but the beauty of EIT is it's invasive, that you have to do an operation and put it in, but it's round the nerve and it's not got anything going into the center of the nerve and damaging it. And the bottom line is, we discovered for the first time that there are fascicles that are connected specifically to the larynx, to the heart, and the lungs, which was a novel physiological finding. And I can just explain the experimental paradigm. So we worked with a large pig facility near us in London. We'd put one ring of electrodes where we do EIT, and then we have the same electrode design, but there's two rings which are about 4 millimeters apart. So we'd have two bites of the cherry. Firstly, we needed a trigger. So if you remember, you need a trigger for the brain activity. You need a trigger for this. So what we do is we record over a couple of minutes to a few minutes and trigger off the EKG. Anything unconnected with the heart would average out, and then you would produce an image like this one, the cardiac one is, for example, is there. And then if you triggered off breathing, you would get a different one, which was there. And we showed that. And then you would have a second bite of the cherry, because having said that's what we think it is with EIT, you then went round and we did modeling to show that if you stimulated two electrodes, if that's your nerve, and you simulate in 1 o'clock, look to see whether the EKG or respiration changes. 2 o'clock, same thing, 3 o'clock. We call that trial and error selective stimulation. That correlated. And then we were able to find for the first time that there are these fascicles which are organ specific. And that means that if you want to treat asthma in a closed loop way with vagal nerve stimulation, we've now got the basics for that. We're okay for time. This isn't directly relevant to EIT, but it's one of my favorite slides. So we've got this stellar who was a PhD student, and she was a biologist, not a physicist. And when we had these findings, we said we want to have an independent validation of this. The fact the two of them matched up was pretty good. And the way to do that is, this is my favorite, you can't see all of it. She's asleep on the floor because we did this heroic experiment, which would be 18 hours long with our veterinary surgeon on the pig. You know, you want to, if you're using a pig, you want to get all the stuff out of it. And at 4 o'clock in the morning, she'd wake up, the pig would be terminated, and she would climb inside it and dissect out the nerve from the neck all the way down to its heart, lungs, or its stomach in recent experiments. How they do that, I don't know. And then she would take it, and we pioneered this technique. So when we started the project, the question is how you could trace these fascicles going up 50 centimeters. And there were 1 or 2 publications on histological sections or using micro CT, but they were only for a few centimeters. And although she was a biologist, she came up this way. You stain it with iodine, she chopped it up into bits, but you remember how the bits all link up, put them in a micro CT, which chunders away overnight. And then at the end, she was able to trace the whole fascicle over 50 centimeters. And this all matched with what we found with our other techniques. You can get a beautiful video like this of seeing how the fascicles go up. Then you can segment them and produce images like this, and then show these are the different colors showing the cardiac and pulmonary. So that all makes sense. And it's now been widely adopted by quite a number of groups. And this was the project that was DARPA and NIH funded. Now it's MRC funded. Now we're translating this to human studies, which we are able to do in our own institution. And we're doing in vivo studies. So we have patients that having vagal nerve stimulators fitted for epilepsy. And we've got a friendly neurosurgeon, like it sounds like you have here. And the poor chap, he doesn't sleep. So if we want to meet with him, I don't know about your neurosurgeon, usually the only time he can make is 7 o'clock in the morning, by which time he's already been round 2 hospitals. And he's not paid to do research, but he's just great. Anyway, he allows us half an hour during this experiment. We've specially made human-safe electrodes by making them more flexible. And we've got an implanted device stimulator, which is also human-safe. And without going into details, we've shown, got the same results as in the pig. And we've also been able actually to show efferent and efferent cardiac fascicles. So that's all going very well. And then we're developing this technology for further human studies. And again, we would like other people to use this, and any collaborating group that's interested would be happy to supply the kit to. So we're down to here, and I'll just 5 minutes more. So we're still on method development to achieve the goal. So this is one thing that we're doing. So you may well be familiar with this audience with MEG, which is magnetoencephalography. I don't know, do you have a system here?
We did up until about 10 years ago. So
okay.
The great years of much remember it. Yeah.
Yeah. So that's done with squids, which require liquid helium. It's amazingly expensive to buy and to run. But in the last decade or so, they've got these at room temperature atomic magnetometers. And the leading company in the world is not far from here in Colorado, called Q-Spin. And that makes a little one like this. They cost about $10,000. And it's just laser interrogation of rubidium vapor. And the idea is the people who do MEG claim that it's much better than EEG, because of course the magnetic signal doesn't worry about the skull, which is the thing that's made our signal to noise compromised. And so we think we can achieve the original dream of doing this with a cycle helmet with atomic magnetometers. Now it turns out, because we need our sweet spot of 2 kilohertz, you can't inject enough current with coils. The ideal would have been to inject with coils and measure magnetically. So we're still stuck with EEG electrodes for injecting the current, but we're measuring with this, and we think this is going to allow us to achieve this. There's a problem. The problem is that there's a lot of really sophisticated engineering and know-how in making these things work, because they're measuring incredibly tiny fields of femtotesla. They're all optimized for the EEG, which is up to 100 hertz. We needed a 2 kilohertz, the story of my life in EIT. Everything we want to do, we have to tailor make for us. So we're hoping that we can tweak the Q-spin one so we don't have to make one. We're doing those studies at the moment, and then work. But if not, we're going to have to make them ourselves, and that's at least 2 years' work. But I'm very proud to say I can now put on my collaboration list that I'm collaborating with a quantum physicist, which I think that's very exciting. Right. Finally, the next one, the thing that is also limiting for anybody that would like to collaborate and use it, is this annoying thing that we have to have a repeated trigger, and it's a few minutes of something repeated. In principle, you could get round this by frequency division multiplexing, and that's by, if we could find frequencies where you get a similar signal at a number of different frequencies, and we can detect it, then you can do the same thing for the fast neural we do for the slow by injecting different frequencies on different combinations. That's work in progress, but our preliminary results are encouraging. This is the sensitivity map for nerve, right sciatic nerve. This goes up to, I don't know if you might not be able to see it there, 100 kilohertz. There's a relatively flat area. Our signal to noise isn't as good as where we measure at the moment at 6 kilohertz, but it's pretty good. And we think if we can go to this flat area, we could then measure all the different frequencies at the same time. And likewise for the brain, preliminary results are, and we were showing this in a poster at in NER just last week. So without going into details on that, we're optimistic that we might still, in my scientific lifetime, achieve the dream of a cycle helmet, show them a picture of their grandmother, and you'll see all the circuits lighting up in real time. And so that's the dream. And then I'm going to skip that one as we're out of time. So in summary, this harebrained idea of fast neural EIT, does it work? Yes, it does work, but not quite in the way we originally intended in the brain, but it definitely works in the nerve or fascicles. And I think it's not unrealistic that in 10 or 20 years, there might be millions of people wandering around with a vagal nerve stimulator, and they're modulating immune function, possibly endocrine function, treating diabetes, and they'll all have an EIT chip in because they'll use that to see which fascicle they want. And this one, that's in, we hope that people in neuroscience, cognitive, animal research will be using our kit. That's ready imminently with depth electrodes, but that would only be for animal research. This one is in amber because this is hope rather than definite, but I think it's not unrealistic that we can achieve the aim of the cycle helmet, imaging those fast neural pathways in the brain. Thank you very much for your interest in it.
At 1.5 kilohertz, is there any information in the real and imaginary difference?
No. The EIT measurement is just beset by artifact. You've got to be so careful that you're not measuring instrumentation errors. So the general rule of thumb is that the main change is actually resistive and real. All the errors you get from stray capacitors, they're fairly minimal at 1.5 kilohertz, but we just look at the real component. In fact, the phase angle is so low at 1.5 kilohertz that we can get away with the modulus. But to the best of our understanding, there isn't additional information in looking at the imaginary component.
You mentioned also higher frequencies.
Then you just throw them away because that's where all the errors are. The bugbear as you go in higher frequencies is stray capacitance, and that translates into errors. You get common mode errors in the imaginary component.
With vocal DNS, say you're doing it to avoid vocal stimulation, is one of the next steps ensuring that efficacy for like epilepsy or depression remains the same? Because like you said, DNS is very unknown why it works. We worked on VNS for depression trial, so
I'm sorry, I can't hear you clearly.
Oh.
Yeah, sorry. Coming in.
If you do focal VNS stimulation to avoid vocal stimulation, do you think it will still work as well for epilepsy or depression? Or are there too many unknowns in VNS?
Great question. So I can't believe nobody understands how VNS in epilepsy works. So, for example, we've got a grant application in at the moment for treating heart failure, where there's good evidence with a UCLA group. We can get the cardiac fascicle. The sort of thing we can offer is to avoid stimulating the larynx. But because we don't understand how epilepsy works, we just got to do that by trial and error. So what we'll be able to do is say we can avoid the larynx, but because we can't find that one organ, the afferent pathway, we don't know which one we want to stimulate. So, and it's similarly for depression. Whereas if it's one of the things that the GSK are interested in, and there's a few other pharmaceutical companies which are efferent ones where there's a particular organ. So, for example, gastric motility, there's a lot of gastric, that could be one. Then it'll be easier for us to identify that and then try and selectively stimulate it. I think one of the potential real uses could be for asthma, in which you would have a closed loop way of constantly adjusting autonomic drive to the lungs for asthma, or possibly another one which we published and generated a lot of interest in COVID was whether if you stimulated the pulmonary fascicle, this could suppress the cytokine storm. In those, we know what to target. So until somebody finds out how it works in epilepsy, the answer is only indirectly.
And so for commercialization, you're going for efferent stimulation first, trying to get it down to the low.
Yes, I suppose so. Yes. Yeah. Good point.
So you mentioned extending this work to neuroscience relevance. Now I'm wondering if this setup exclusively works for like ethics animals, or if it would be compatible with like doing
For humans as well was the question.
no, sorry, for rodents, like in rodents, if you're using this for a neuroscience study, you have to fix their head and then put them on a treadmill, or
No. So at the moment we're not doing it with a portable, but we sourced one. It could just be EIT can be made very small. We've actually got Samsung half-made a whole EIT system on a chip. No, in theory, you could have something with just a little portable transmitter on their neck, and they could run around and be freely moving. And it would be able to, in those sorts of experiments, it would be able, if you could put in these subdural mats and leave them for months, and then you would be able to image everywhere in the cortex whilst they do quite complex tasks.
with the subdural depends with the signal quality,
Sorry,
does the signal quality degrade significantly over time with the subdural?
Over time?
Yeah.
We don't know. We haven't done any. All our experiments have been acute so far, but with the subdural mats, probably not. With intracranial ones, that of course would be a worry, just as they are for U2 arrays when they go in. If there's going to be, we're reliant on there being a low electrode impedance into the tissue, so it could be a problem when we do the penetrating ones, but we haven't done that yet.
So, I think we're like notice any differences in ball depending on the type of stimulation that is applied, like you're vertically applying it, or whether it's something that's coming from their own sensory, or feel like maybe a difference in quality, or.
Okay, so all we've done it in so far is whisker movement, sensory, and visual. And the answer is no. We don't know exactly what the biophysical basis is. We biophysically model what the changes are in nerve, but we don't know what proportion of the cells are depolarizing, and we just know empirically that we get a signal of a certain size. They are all actually, no, that is a good point. They're not all similar. SEPs I like because you get an intense, very short activation of S1, and that gives us a nice synchronized signal. And likewise, the whisker one as well gives us a big signal. Visual evoked responses, the area under the curve is about the same, but it's much more spread out just because the visual evoked response is a longer latency and is more spread out. So the signal to noise on that one is more challenging. But I think what we've seen so far is that empirically, whatever is going on, the same proportion of neurons and depolarization. And the other thing is we don't know what proportion is actually action potentials and how much is graded depolarizations in the dendrites. I would love to do the study where we model that and we take what was a whole PhD just modeling this in nerve and do that on the complex geometry of the brain. But we haven't done that project yet. Yeah, thanks.
Are there any other? No, I'm in the chat. Okay. All right, thank you, David.
Yeah, great.
Pleasure.
So my dad, he goes home and line.